So I'm Dr. Stephanie Cole, and as part of my job on the product squad, I speak to a lot of researchers working in the biologics field. Over the years, I've learned a lot of your most common concerns and challenges in the lab. And for any of you who've ever worked to optimize some sort of protein-based therapeutic or target, you're likely already familiar with the use of melting temperatures, or TMs, to rank candidates. I spend a lot of time working on making the tools that measure TMs, along with other stability parameters. And I also work with researchers to learn how to improve these tools available to them in order to do this thermal stability characterization and work more efficiently. So today I'd like to deep dive into thermal stability. It's a concept that in some ways is taken for granted. So I really want to discuss why it's such a common method, what different approaches are available for measuring it, and some of the limitations that it has in biologics drug development. And then after that, I'm going to show a few examples of how it's used to select conditions and drug candidates that are more likely to make it to market. Now, if you're working with protein-based therapeutics, you may already be familiar with the concept of developability. Developability work, or building a developability profile, means collecting a lot of information about your constructs, often in different buffer conditions, in order to determine which candidates and buffers optimize the stability of the therapeutic best for its long-term development and manufacture. Now, there are a lot of parameters used to create a full developability profile for your candidates. Please check out the link to the video below, how biologics teams approach developability screening of their candidates with combinatorial stability characterization, if you'd like to learn more about those different parameters that you would use to create a developability profile. But here's an example of the many quality attributes that researchers look for in their candidates. As you can see, it is a huge list. And while I'm going to focus on thermal stability today, it's worth noting that one or two parameters is never enough to get a full picture of your candidates. There is a pile of data to collect using lots of different assays and instrumentation. And while you have to put all of that information together in order to make decisions, rarely does one candidate top the list in every category. But by understanding where each piece of data comes from and what it tells you, you'll be better prepared to prioritize candidates and select ideal buffer conditions. All right, so thermal stability parameters reflect how well your protein remains folded in response to a temperature denaturation gradient. Stable, active proteins are well folded as their form dictates function. So now when heat is applied to the system, what you'll see is that proteins unfold and will lose their activity. The melting temperature is found by following some aspect of protein folding and plotting it against the temperature of the sample. The TM is the point at which 50% of the protein is unfolded and 50% remains folded. We'll talk in a moment about the different approaches that exist for determining this value, but very generally, a soluble, stable protein unfolds at a higher temperature. Therefore, the TM, or melting temperature, is used as a ranking parameter to say which proteins are the most thermostable and therefore better candidates for progression. Now, thermal stability is a popular ranking parameter because it is very quick and easy to measure compared to most of those other developability characteristics that I showed you earlier. Now, there are different methodologies for finding TM, which we're going to discuss a bit in detail, but all of them require fairly low sample volumes and collect measurements within an hour or two. And compared to setting up many of the other developability assays, such as activity assays, probing post-translational mods with mass spec, finding a TM is just so much faster and simpler compared to a lot of those. And as a result, it has become a go-to method for ranking parameters, and in particular for eliminating the worst of your candidates. If you've got dozens or even hundreds of candidates and conditions to comb through, having a strategy to dramatically thin out that herd early on is going to save you a lot of time and effort later on. Now, it's worth acknowledging that there are questions about how good of a stability indicator thermal stability parameters are. Biophysics researchers have spent a lot of time trying to correlate a melting temperature value to the absolute stability of the protein, and there is rarely a clear-cut correlation. This is another reason why looking at all of those developability attributes at once is so critical. Thermal stability is a handy indicator of a stable protein-based therapeutic, but there are many other factors to consider that encompass the broad category of stability. Colloidal stability, chemical stability, and overall activity are all critical quality attributes that must be examined in order to make the best possible decisions about which candidates are worth progressing. However, the thermal stability experiments I'm about to describe are fast and easy, and they allow you to make changes to either your candidate's primary sequence or their buffer environments and really quickly identify which of those changes increases the thermal stability. So while it may not be the whole picture, it does have low material requirements and can be done quickly. And because the data is pretty clear-cut on the interpretation, thermal stability profiling remains a very popular and viable approach to early candidate triage and selection. All right, so let's get started talking about the different approaches available to you to measure the melting temperatures and the pros and cons of each. The first method we're going to discuss is DSC, or differential scanning calorimetry. This method has been available to researchers since 1962 and was used for several decades as a go-to method for determining the TM values. And as a result, melting temperature values from DSC are often considered the benchmark against which other techniques are compared. Now, DSC works by applying a steadily increasing temperature gradient to a reference sample, the buffer, and an experimental sample, the buffer containing the protein of interest. Now, each of those samples is going to heat up in response to the increasing temperature. The reference sample has a known heat capacity. The experimental sample has an unknown heat capacity, and that difference is compared against the reference. The goal is to keep the temperature of both those sample volumes the same throughout the experiment. And there's basically a blip in the heat capacity near the unfolding transition because of the energy required to disrupt the tertiary structure of the protein. In other words, protein unfolding has an impact on how the experimental sample reacts to the change in temperature. The phase change associated with folded to unfolded requires more heat to maintain the same temperature as the reference sample. That amount of extra heat required, and the temperature at which the phase change occurs is recorded, which then gives the melting temperature and the enthalpy of unfolding. So DSC is considered the standard for TM measurements, not just because it's been around for so long, but because it works in so many different fields. Polymer chemists, liquid crystal researchers, and chemical analysts all use DSC to do their work. DSC instruments are designed to be highly sensitive, and many function at a wide range of temperatures. For biologics researchers, though DSC does indeed give highly reproducible, thermodynamically precise melting temperatures of therapeutic proteins, it does have some downsides. The need for a reference sample for each experimental sample means preparing twice as much for any experiment. Further, most DSC instruments aren't really designed with high throughput in mind and just have the one space for the reference versus experimental sample, which means each experiment has to be done one by one. Now, while the sensitivity and temperature range of DSC is a huge boon to material science researchers, it's actually less necessary in biologics. It's not really needed to profile a biologic outside of 20 to maybe 100 degrees centigrade, and very rarely are researchers working with such low sample concentrations that it's difficult to tease the signal from the noise. DSC instrumentation also scans each degree change in temperature slowly, meaning it can actually take quite a while to run a single experiment, and since you don't have high throughput, it ends up taking you a long time to gather all the data you need on every single candidate or condition you're screening. So now taken together, it's easy to see how and why DSC has been so valuable in the past. However, with the current needs of the industry being geared less towards the high thermodynamic sensitivity and more towards getting results in a high throughput manner, DSC is less popular of a technique for determining melting temperatures, particularly during these early developability, profiling, or candidate selection processes. So now I'd like to talk about a technique called circular dichroism. Circular dichroism, or CD, is a spectroscopy-based methodology for assessing the secondary structure of proteins or nucleic acids. I don't want to get super into the physics of light and the chirality of biological molecules. So very, very simply, left and right circularly polarized light goes into a sample, and the rate at which the left and the right polarized light comes out is impacted by what is inside the sample. The secondary structure of proteins, namely their alpha helices, but also some other elements, is going to impact the left versus right circularly polarized light differently, and that difference is measured by the system. So to find the melting temperatures with that information, the CD spectra is collected as a function of temperature. Then algorithms deconvolute the information to find the enthalpy of unfolding and the melting temperature. So CD is super valuable because it also provides this basic insight into the secondary structure of a protein. There are typical fingerprints for many different secondary structure elements that can be pulled from the data. So it gives you sort of that additional insight into how your protein is physically shaped. Now, there are CD instruments out there that are higher throughput, capable of running 96 12 plates multiples in a row. And even though each scan is executed in a few seconds, getting high enough sensitivity to confidently calculate a TM requires at least several dozen scans for each sample, which do ultimately add up in the amount of time it takes to calculate the data. So while you can run more samples at once without having to restock your instrument, it still takes time for those experiments to run. Additionally, the secondary structure information, while very cool and helpful for certain proteins, like those without NMR, crystal, or cryo-EM structures, it's kind of extra information that isn't always required to actually make the decisions about candidate selection. And finally, another drawback is that CD is highly sensitive to common buffer additives such as sulfates, phosphates, and acetate, unless they are highly dilute, which limits its use for many typical biologic buffers or for formulation screening. I'd like to talk now about DSF, or differential scanning fluorimetry. There's two different approaches I'm going to cover within this bucket. The following approaches both use spectroscopy to measure protein unfolding. I'm going to start with extrinsic fluorophores. For protein unfolding applications, the most commonly used extrinsic fluorophore is called Cipro Orange. So I'm mostly going to contextualize that approach using that dye, though there are others out there that exist. So with the advent of qPCR spectrometers, which are a staple in pretty much any biomedical research lab, researchers turn to this common multi-purpose instrument to find a way to determine melting temperatures. And what they came up with was Cipro Orange, which is a dye that fluoresces in range of most qPCR spectrometers with an excitation peak around 480 nanometers and an emission peak around 570. So what's cool about Cipro Orange is that it preferentially binds the hydrophobic region of a protein. And since the hydrophobic regions are usually buried when a protein is properly folded, when that extrinsic DSF experiment starts, there's going to be almost no fluorescence detected. And then as the system heats up, the protein starts to unfold, and the hydrophobic regions of the protein become exposed to solution. The Cipro Orange dye binds these regions and lights up. Now, the melting temperature is deconvoluted from this information by looking at the transition from unbound to bound from the dye as determined by the increase in its fluorescence emission. So the great thing about extrinsic DSF is that it can be done in a very high throughput manner with 96 or 384 well plates. Experiment time varies, but generally a temperature ramp of 1 degree C per minute is reasonable, which means that a full experiment is done in about an hour and a half. There's some preparation required with the dye, but liquid handlers can help make short work of the experimental setup process. So while qPCRs are standard equipment in many research labs, and while Cipro Orange dye is inexpensive, there are some drawbacks to consider with this method. First, since Cipro Orange is sensitive to hydrophobicity, its fluorescence increases in detergents, making it impossible to see a transition stand out when the protein is in a detergent-containing buffer. Now, while low concentrations of stabilizing detergent like Tween may not be enough to interfere, if you're thinking about screening stabilizing detergents for membrane proteins or something intrinsically disordered as an antigen, it would not be possible to buffer optimize these systems. Furthermore, if you happen to be working with a protein that has a hydrophobic patch initially exposed to solution as just part of its structure, that's going to light up the dye really early and possibly remove the ability to see the transition to unfold. Additionally, since that extrinsic dye is essentially a small molecule, there are concerns that by adding it to the sample, you're stabilizing or destabilizing the protein of interest because it can artificially bind the protein. This will result in a shift in the TM value that's actually not related to the construct or condition you're profiling. And often it's only able to see one transition with separate orange, as the dye can quickly saturate the detector after the first domain unfolds. So if there are multiple unfolding events occurring, it's not always possible to see. Okay, now I would like to discuss intrinsic fluorescence. I'm mainly going to contextualize this with using nanodifferential scanning fluorimetry, or nano-DSF, a technology for monitoring the unfolding of a protein using its intrinsic fluorescence. It's a technology that's unique to our instrument, the Prometheus Panta, which was optimally designed for the most efficient heat transfer possible and the most sensitive detection of those wavelengths. However, the general principles behind how to use a protein's intrinsic fluorescence for measuring thermal stability are similar, even when we're not talking about such a specialized spectrometer. Now, tryptophan residues have different emission maxima depending on whether they are exposed to solution or buried in protein. And since they're hydrophobic residues, when a protein is folded, they are generally buried in the protein core. And in response to a temperature denaturation gradient, they become exposed to solution. That emission maxima shifts from 330 to 350 nanometers. And with highly precise optics and direct heat transfer, we can closely monitor that shift and identify the inflection point of this transition, which is the TM. I want to point out, because I'm going to show some nano-DSF later on, it's often easier to see these inflection points by using an algorithm to determine the first derivative plot. The inflection is at the peak of the first derivative. Here we see both the T onset, which is another really useful stability parameter. And we also see the two melting temperatures associated with different domains unfolding in this antibody example. So, like I said, not only do you find the TM, but you can also find the T on or onset of melting. The sharpness of the transition is another indicator of stability. A sharper transition from folded to unfolded is a more stable protein compared to one that starts unfolding slowly at a lower temperature. You can look directly at the slope at IP or do a general estimate by looking at how close the T on is to the TM. The closer in value they are, the sharper the transition. Again, this is reflected by a very sharp peak in the first derivative rather than that broader peak you see in sample B. So since nano-DSF uses intrinsic fluorescence, it's buffer independent. The raw fluorescence values at 330 and 350 don't matter per se, as it's only looking for the shift in fluorescence that occurs upon the unfolding transition. Therefore, if you have background fluorescence at either wavelength, as long as it isn't heat sensitive, it's essentially subtracted from the reading once you're looking at the ratio. This makes nano-DSF great for anyone looking to do buffer screens or thermal shift assays. You're free to screen as much detergent or other hydrophobic or small molecules as you'd like. Nano-DSF requires 10 microliters of sample and can be done in a high throughput manner. Experiments can be run in as low as 15 minutes, and you can run up to 1,536 samples without going back to a load. This means you can generate a lot more data a lot faster than most of the other methods. And unlike the extrinsic DSF approaches, you don't have to worry about the false stabilization effects from the dye or missing additional transitions from other domains. Finally, it's worth noting that the nano-DSF in Prometheus Panta in particular provides high-resolution data. The heat is transferred with maximal efficiency to the system, which means there's no delay between the reading and the system reaching the assigned temperature, giving it the highest resolution of these optical methods for thermal stability measurements. You can even see transitions in samples with concentrations of protein as low as 10 micrograms per milliliter. Okay, now we have talked all about the different approaches you have for finding melting temperatures in order to rank your candidates, but I want to look at some real-life examples. Once again, I'm going to pull these examples from nano-DSF data specifically, although many of these experiments are possible with the other methodologies. So let's first look at an example of how TM measurements are useful for buffer screening. This is an example of an antibody in 6 different buffers from pH 4.0 to pH 9.0. The buffer salts are kept at 25 millimolar, and every sample also contains 100 millimolar of sodium chloride. Now, as the pH gets lower, that first transition, the first melting temperature, is decreasing. And you can also see that between pHs 7 and 9, they all have really similar melting temperatures. They also have similar slopes at the inflection point, and the T-on values are all about 5 degrees before the melting temperature. Ranking solely by TM, the higher pH values are more stabilizing for this antibody. However, I want to take a moment to offer a word of caution here because this is a really good example of why thermal stability is not the whole picture. I want to talk briefly about colloidal stability. So I'm going to take a little detour here. Please bear with me. We can monitor the turbidity of a sample, which is basically aggregation, in parallel while running these melting temperature experiments. So as large aggregates form, they reduce the amount of light that returns to the system, which again is an indicator of the colloidal stability. Now, if we look at the degree of the turbidity increase, as well as how early on in that melting the turbidity starts, we get another indicator of stability alongside that conformational information we get from the thermal stability experiment. Higher temperatures of turbidity onset indicate more stable samples. So with that in mind, I want to go back to that data, but I'm going to add the turbidity measurement on the same samples. Now, what you see here is that actually when we go down to pH 4, it prevents amorphous aggregates from forming, and there is essentially no onset of turbidity, and it doesn't reach saturation. Of course, with the low TM1 that we saw from the nano-DSF information, we may actually kind of want to tread the balance between these. We see that the pHs 5, 6, and a little bit of 7 have a higher onset of turbidity compared to those higher pHs 7 through 9. So with this in mind, the optimal formulation might actually be somewhere around pH 6. So I just wanted to point that out to remind us the importance of having multi-parameter screening when we're doing our developability work. So next, I want to talk about thermal shift assays. These assays are helpful tools to quickly screen for binders. When a small molecule or other ligand finds a protein of interest, it generally stabilizes the protein thermodynamically, though in some cases, it may actually see a destabilization. So here we're looking only at the first derivative view of several different compounds in complex with a protein. AGP is the protein of interest, which is the black first derivative curve on this graph. Its unfolding temperature is around 64 degrees Celsius. A DNA encoded library was made, and then assorted molecules are small molecules derived from this library, and those are the different numbers you see here. Compound 22, shown in red, is the most stabilizing. It offers a 5.94 degree shift in the melting temperature. And then finally, I'd like to go and talk about GPCRs. These are really important targets for a lot of diseases, and it's often important to be able to purify and stabilize them in order to do research on different diseases and mechanisms. So nano-DSF is really powerful for a wide variety of potential proteins of interest. This GPCR was purified out of the membrane and not solubilized using nanodiscs, so it needed substantial detergent in order to remain soluble in solution for further assays. By using the intrinsic fluorescence of nano-DSF, we could actually do a stability screen for the buffer detergent additive in order to find which was the most stabilizing. So with that, the information about thermal stability is a really fast and effective way to select candidates or buffer conditions that are more likely to remain stable long-term for assay development, further characterization, or storage and delivery. While thermal stability measurements aren't the complete picture on protein stability, the ability to conduct screens in a high throughput manner using low sample volumes make them a super valuable tool for anybody looking to screen libraries of engineered proteins, buffers, or small molecules quickly and efficiently.